So now we will hear from Dr. Wolfgang Köhler. He's going to go through some dietary management, which is pertinent to ALD and AMN. Thank you.
Yeah, thank you very much, Karen. Thank you, ALD Live. Thank you all. I'm very happy to see you surviving last night dinner as a very long-chain fatty acid challenge. And I know why you urged us to build a paper and straw tower. And that was a precursor of this here, right? Yeah, okay. Well, oh, let's see. This one, yes. Mm-hmm. Okay, so I got the bad part of it. Dietary treatment. Who of you is making dietary treatment for XALD? Raise your hands. Oh, at least two. Yeah.
In the past.
In the past.
Not now.
Well, not now. Yeah, that's kind of a problem, right? Yeah. Okay, but I'm still here to remind you that this disease is in fact an inborn error of metabolism. So what we find in all of the patients with this disease, there is an accumulation of these substances called very long-chain fatty acids. And this is diagnostic on the one hand side, but also the question is, what does that very long-chain fatty acids do in your body? So this is that's what I want to discuss with you in this talk. Well, very long-chain fatty acids, well, look at this, it's very complicated. We are talking about fat, simply fat, and it looks so complicated. We have different kinds of fats in our body. There are very long-chain fatty acids, they are saturated. Saturated means that the carbons there are long chains of carbons you can see here, they have no double binds. They have 4 binding places, and there's something different at all of these 4 binding places, and no double binds. And then you have mono unsaturated fatty acids, you have 1 double bind, and you have polyunsaturated fatty acids. These are terms that you hear quite frequently in the advertisements of some wonderful butter, which is full of polyunsaturated fatty acids, for example. And maybe you know now what it is about. So some of these fatty acids are essential. What does that mean? Essential means that your body is not able to produce them by themselves, by himself. So you have to eat this. This is why it's called essential. And there's a second thing I wanted to tell you. The rest of the fatty acids are produced by our bodies internally. So they are produced from shorter chain fatty acids to longer chain fatty acids using so-called elongases. This is important for the disease ALD. The elongases are enzymes that elongate shorter fatty acids to longer fatty acids. So keep this in mind, we talk about this later. So where do the very long-chain fatty acids come from? Well, they come firstly from that what you eat, from the food. For example, last night we ate big amounts of very long-chain fatty acids. You were full of it today. But if we haven't eaten any very long-chain fatty acid last night, our body would be able to elongate very long-chain fatty acids from shorter fatty acids in a structure called endoplasmatic reticulum. You've seen these pictures already, and you see in this factory in our body, which is present in every cell, in every body cell, you find such a ER factory, that is elongating long-chain fatty acids. And you see the workers here are the elongases, which elongates shorter fatty acids to longer chain fatty acids. So this happens every day in our body, in every cell. Why is that? No, that's why we need very long chain fatty acids. That they are important for our body. They have essential physiological functions. For example, the myelin maintenance is primarily, or is very much involved, that very long chain fatty acids are needed for this myelin maintenance. They are incorporated in substances like this, for example, and these substances are major components of biological membranes. So they are incorporated in these membranes. That's the normal situation, and that's why we need very long chain fatty acids in our body for the myelin function. But what happens if this too much very long chain fatty acids are in our bodies? So then you may have some diseases, for example, a disease like this, where there's a clear-cut enzymatic defect in this disease, and you see a leukodystrophy coming up. So these substances, which are normal in our myelin structures, they have unique biological properties, and they are important for the membrane stability. They're important for the function of the myelin. So if they are disturbed, there may be some disease. For example, if there's too much very long chain fatty acids, then there may be some disease. So the question is, what is the role of very long chain fatty acid incorporation XALD? Since we know we have much too much very long chain fatty acids in our body. So in all ALD patients, there's a biochemical defect which is caused by an enhanced fatty acid elongation on the one hand side, and on the other hand side an insufficient fatty acid degradation. So in the result, we have an accumulation of very long chain fatty acids in every patient with ALD. So, and what we can see that there is a high, there's a 39-fold excess of very long chain fatty acids in some compounds that are really, really necessary for a good myelin function. They are selectively, we can found these elevated very long chain fatty acids in active demyelin lesions, for example, of the brain of ALD patients. So in these active demyelinating lesions, we see a lot of very long chain fatty acids accumulating in these lesions. We can see this, for example, in crystalline needles and in the microscope. And this elongation of very long chain fatty acids is causing brain dysfunction, for example, a lot of cellular stress. We also can measure this high concentration for newborn screening, for example. We measure this compound here. And so what happens with this elongation is there's a lot of pathology going on if there's too much of very long chain fatty acids. And a colleague of ours from Vienna put this all together, saying that elevated very long chain fatty acids have a chronic toxicity of brain structures, leading to some energy depletion and energy problems in the mitochondrium, and leading to oxidative stress and leading to disturbed myelin interaction with the axon. So loss of axonal support and axonal function is caused by chronic toxicity of very long chain fatty acids that's finally leading to a chronic disease of the long axons of the nerve cell. We call that axonopathy, and the clinical symptoms. So there's a direct correlation between chronic toxicity of very long chain fatty acids and disease. right? Very importantly, recent research showed also that these are, for example, long-chain fatty acids. They incorporated in the mitochondrial membrane. Mitochondria are are needed for energy supply for a body. So if they are incorporated here in this mitochondrium, then the energy situation of the body is not correct. So you have less energy, which causes a lot of stress. We call this oxidative stress. And we can measure this. You can measure this in the body. You can also measure it in some animals. And these species that are coming out, which are toxic for the body, caused by the oxidative stress, will then secondarily cause axonal damage and finally death of the axonal. And our intention must be that we can stop this process. We need to stop the oxidative stress as early as possible for potential recovery of an axon. If it's like here, it's irreversible. You can also see this in the neuropathology in XLD mice. These are normal myelin sheaths. What you can see here, there's an axon, and this is the myelin sheath around the axons. You see, this is a normal situation. And in the ALD mice, which are diseased by XALD, you see a swelling of this myelin, and later on there's a destruction of the myelin. So we have a model, in an animal model, where we really can see what happens if long-chain fatty acids keep right up all the time during their life. Of course, we are, in these animals, in the mice, we are trying to reduce this oxidative stress with substances like pioglitazone, for example. This is an experiment from Barcelona group, showing that using this medication in the mice, not in the human, is able to prevent axonal damage in these mice. You can even see this in the mice itself. Or not. Can you do something? Ah, forget it. I wanted to show you the two mice. That's very nice. It's not working. Doesn't matter. So, well, it was a bit complicated, but you need. I need this information to provide to you, that you may understand what the background is of all this. Yeah. And the current pathogenetic concept of X-linked adrenoleukodystrophy, it's not only the gene defect the gene defect is directly leading to pathology, to axonal degeneration, to disease, to inflammatory demyelination, but also the gene defect is leading to elevated long-chain fatty acid, which causes oxidative stress and energy failure, and also causes neurodegeneration, and finally leads to some clinical symptoms. So it does make sense that we focus on very long-chain fatty acids to reduce this substance, because it's causing a chronic toxicity. Every patient has this long-chain fatty acid measurable in plasma, and we know that these elevated long-chain fatty acids are neurotoxic, they are pro-inflammatory, they are initiating oxidative stress, and they are inducing neuronal death. So these substances clearly are to be focused in any kind of treatment. Well, the background of the treatment is that you see an excess of long-chain fatty acids in all tissues that are involved with this disease. You see that in the brain. You can see that more in the active brain demyelinated lesions compared to normal appearing white matter. You see an increase in the brain of the various phenotypes. There's a difference. the more severe phenotype to have more increase of very long chain fatty acids. And we also know that very long chain fatty acids, when you expose these substances to brain tissues, this may be followed by an inflammatory reaction. I think Florian actually had some experience on this. You see a direct combination between elevated long-chain fatty acids and inflammation also in experiments. And also there are experiments from a German group showing that if you expose normal brain cells to very long-chain fatty acids, they will, in culture, in a cell culture, in an experiment, well, 24 hours later, there's no cell left. All cells are died. So they are really toxic to neuronal cells. We know that long-chain fatty acids elevated in animals, that's experience from animal experiments, will lead later on to clinical symptoms in the mice, which are similar to that what we see in AMN. There are also other animals, for example fruit flies, that have some pathology caused by very long fatty acids, which are reversible by treatment with Leukodystrophy oil, very interestingly, but these are very old experiments already. And we also know that an overcorrection in animals, at least in mice, of metabolism of very long chain fatty acids may correct also the clinical symptoms. So, GTE therapy or Leukodystrophy oil therapy as a mixture, 4-to-1 mixture with GTO and GTE is able to normalize very long chain fatty acids within 4 to 6 weeks. Every patient has normalized very long chain fatty acids. Well, the effects proposed in a recent study saying that there is a direct effect on the elongases. You remember the elongases are enhanced in this disease. So the Leukodystrophy oil has a direct inhibition effect on these elongases, leading to less producing very long chain fatty acids on the one hand side, and there's also an indirect inhibition with a competitive inhibition. So these are experiments very recently showing that this treatment really changed something in your body in terms of normalizing very long chain fatty acids. So the hypothesis for the treatment is these substances are toxic, so I don't want to have the toxic substances in my body. So the intention of the treatment is to normalize very long chain fatty acids. Well, this nice gentleman, Hugo Moser, showed us many years ago that treatment with Leukodystrophy oil is really normalizing. This is the normal range of very long chain fatty acids, and this is the start of the treatment with Leukodystrophy oil. And he can show that that treatment with this Leukodystrophy oil is normalizing the very long chain fatty acids in your body. However, we had a series of smaller open studies with this treatment that show that there is no fully protective or preventive effect of this treatment clinical-wise. However, there were some critical points with these early studies. All of these studies, or many of these studies, has completely inadequate measures. For example, visual evoked potentials as an outcome measure is not that what you want in AMN patients. They had all small numbers, they were all very heterogeneous, and the most important, they are non-controlled studies. None of these studies had a control arm, so it was simply not, you cannot say anything out of these studies, unfortunately. There's only one study which is controlled, but an open study, which is here. This is a a study in patients where the treatment was started before onset of symptoms, before the age of 6 in children, and the main outcome measure was the onset of symptoms. So this is a very easy outcome measure. You can really say, yes, there's an onset of symptoms. So this is done in more than 100 pre-symptomatic patients where the treatment starts before the age of 6. And then in 2 groups, in the US group and the European group. And there are 2 methods in the outcome measure. The first method was that this group of patients was compared in terms of onset of symptoms to a natural history cohort, which is sometimes very difficult. And the second method is that this group of patients, of the 105 patients, was subdivided in 2 groups. One group had normalized levels of very long-chain fatty acids, and the other groups failed to normalize very long-chain fatty acids. So we have 2 groups of boys, the ones who have normalized, and the others don't have normalization of very long-chain fatty acids. So this is the first comparison in the 2 groups, US and EU. And these are historical controls. And these are Kaplan-Meier curves. Going down is an onset of disease.